All right, so last time left off after bringing up the B1 B cells and how they create or produce natural antibodies, which are antibodies that not only promote homeostasis but are also capable of opsonizing common bloodborne pathogens. And they make antibodies that are frequently reactive to multiple different pathogens. So it's sort of like innate, but we consider them adaptive immune cells in this class. Even though you technically don't need to be boosted or be exposed to the pathogen for them to show up, we still consider them adaptive. Okay. And we left off before I could get into the different types of T cells that there are. Now these become more important later into the class when we learn about T cell development. And you could say the same about B1 cells too. So the major kind of T cell that we care about in immunology is the alpha beta T cell. And it's named alpha beta because of the structure of its T cell receptor. It's got an alpha chain and a beta chain. And these are just the classic T cells. They're CD4 positive or they're CD8 positive. These are the cells that we all know and love. And the CD4 positives, they provide T cell help in the form of cytokines. And the CD8 positive T cells, they kill infected cells. There's another kind of T cell population called the gamma delta T cell that has a different T cell receptor. Okay, so their T cell receptor is distinct from that of the alpha beta T cells. They are not MHC restricted, so they don't respond to MHC, but some of them respond to CD1 proteins that have been loaded with lipids. So CD1 is another antigen presenting molecule, and there's going to be, I guess, more info on this for exam 2. But gamma delta T cells, they aren't usually CD8 positive. A very small number of them are CD4 positive, but frequently they have neither of these as their co-receptor, or they don't express them at all. But they do express CD3, which is how we know that they are T cells. Surveillance, one of their functions is the surveillance and production of growth factors, cytokines, and chemokines in the skin. So they can help to maintain homeostasis. They help skin, I guess, grow at a constant rate by providing skin stem cells with growth factors. They also make cytokines and chemokines in the event of an infection. Okay. For example, there's a population of gamma delta T cells that secretes IL-17 at mucosal surfaces. There's a subset that produces an IL-4 and interferon gamma in the lymph nodes, the spleen, the lungs, and the liver. And there's a subset that produces interferon gamma in the spleen, lymph nodes, and the intestinal epithelium. So they are often found in the intraepithelial lymphocyte population, which is basically just a collection of cells that are found between your epithelial cells in your intestine and even in your skin and other mucosal surfaces as well. So they maintain homeostasis, but one of their major jobs is to provide a very rapid response to an infection. So they are one of the earliest sources of cytokines. We consider these to be adaptive cells in this class, but in all honesty, their function falls somewhere between innate and adaptive. Okay, and then we have the invariant natural killer cells, or excuse me, the invariant natural killer T cells, or the iNKTs. And these are a cell population that has a phenotype that's somewhere of a mixture of T cells, specifically CD4 T cells, and natural killer cells. They express a receptor called NK1.1 that is found on natural killer cells, and they express an invariant T cell receptor alpha chain. So every invariant natural killer T cell has the same alpha chain for its T cell receptor, and a beta chain that has very limited variation. So this tells us that they respond to probably similar molecules, right? They have a very limited repertoire of antigens that they can respond to. It turns out that their major job of the invariant natural killer T cells is that they recognize CD1 molecules that have been loaded with lipids. So just like how gamma delta T cells can respond to CD1 protein, so too can the iNKT cells. Okay, and they secrete IL-4 and interferon gamma. These are an important cell class for defense against pathogens like Mycobacterium tuberculosis, and this is a bacterium that has an extremely hydrophobic cell wall. We're talking like 40 carbon atoms in a single compound in the cell wall of this bacterium. So extremely hydrophobic, very long chain hydrocarbons in the cell wall. And so that makes Mycobacterium tuberculosis kind of a bad fit for the MHC pathway, right? Because there's so much of a lipid coating there that it inhibits the ability of macrophages and such to load anything into MHC. So this is a compare and contrast table for the esoteric cell types. There's the B1 B cells that make natural antibody, for example, against the pathogen Streptococcus pneumoniae, which is an encapsulated bacterium. They cannot be boosted, meaning if you vaccinate, you aren't going to increase the ability, you aren't going to increase the magnitude of the B1 cell response, okay? So you don't expand the response when you stimulate these cells with antigen. And then, oh, and of course, B cells, they don't respond to MHC, so don't confuse that. B cells do not respond to MHC. And in fact, B cells actually express MHC proteins so that they can present antigens to T cells. Epithelial gamma delta T cells, or just gamma delta T cells, they produce cytokines rapidly in response to infections at epithelial barriers. Their ligands are often MHC class 1b, which is sort of a weird subset of MHC molecules. We'll talk more about those on exam 2. And again, they can't be boosted. So vaccinating does not have an effect on the magnitude of the response from the gamma delta T cell population. And then there's the invariant natural killer T cells. These are also a source of rapid cytokine production. Their ligands are CD1 proteins that have been loaded with lipids derived from pathogens. They cannot be boosted. So for all of these different cell types, they can't do one of the most amazing things that the adaptive immune response can do. They cannot give you memory. They don't give you a memory response. So when you're exposed to the same pathogen again, you have the same magnitude of a response against it when it's coming from these cells, okay? One last note before we move on from the basic cells. People in relevant fields to immunology, they divide white blood cells into a few groups. All white blood cells are also called leukocytes, okay? Those are the same thing. Leukocytes can further be divided into two categories, lymphoid cells and myeloid cells. Lymphoid cells are T cells, B cells, and innate lymphoid cells. And of course, natural killer cells are a type of innate lymphoid cell. All of these are derived from the common lymphoid progenitor, and they start out in the bone marrow. You could also call these lymphocytes. They're lymphoid cells, they're lymphocytes. And then the myeloid cells, they're derived from the common myeloid progenitor. Myeloid cells are then further divided into the granulocytes and the mononucleated cells. The granulocytes, those are your basophils, eosinophils, neutrophils, and mast cells. Okay, just like I said earlier. And then the mononucleated cells, those are the macrophages, monocytes, and dendritic cells. So this is also what you would think of as the mononuclear phagocyte system. Also, myeloid cells do include the platelets, which are clotting cells, and the red blood cells, which are also called erythrocytes. Okay, and that concludes this slideshow. And if you have any questions today, unmute yourself for me, because I only have one screen that I'm looking at. Okay, so the next topic that I like to talk about is cytokines, because they're really quickly a source of confusion for everybody. So I feel like I just do it justice to talk about them next. So macrophages are one cell that is an important source of cytokines. Macrophages, they make 5 major cytokines during the response to a pathogen. Okay. They make interleukin-1, which is an inflammatory cytokine. It activates vascular endothelial cells. It causes them to make more adhesion molecule and to contract so that there is an increased gap size between the endothelial cells. It activates lymphocytes, mostly T cells. T cells are activated by IL-1. Okay, it helps co-stimulate T cells. It causes local tissue destruction, and part of the reason for that is because during inflammation, the cells that are leaving the blood to get to the tissue have to break down everything that's in their way in order to get to the site of the infection. So they're going to be breaking down things as they make their merry way to the source of the problem. And it increases the access of effector cells, meaning white blood cells from the blood are better able to leave the blood, traverse the endothelium, and enter the tissue. Okay. And systemically, so those are local effects of IL-1 on basically neighboring cells. That's paracrine effects, right? But then there's also systemic effects. Frequently, systemic effects can be deleterious, so they can be negative. For example, IL-1, if it's released in high enough levels of the bloodstream, it can promote shock, and shock can kill you. But it also has a few protective effects as long as it doesn't go overboard. For example, it can induce fever. So IL-1 circulates through the blood, and it circulates all the way until it reaches the brain. And the brain has the hypothalamus, which is the major control point for a lot of homeostatic functions, right? IL-1 is going to act on hypothalamus cells and cause them to secrete an eicosanoid called prostaglandin E2. And all that prostaglandin E2 does is it raises the temperature set point of the body. So IL-1 acts on the hypothalamus of the brain to tell it to increase the temperature set point of the body. And it does that by making prostaglandin E2, which binds to prostaglandin E2 receptors on muscle cells, fat cells, and the liver, and probably some other cells. And it basically, prostaglandin E2 tells those cells to burn energy doing nothing. So you begin mobilizing fats, you begin burning carbohydrates, and you begin burning proteins. And all of those things help to increase the temperature of the body. Okay, so you begin burning energy. And that energy isn't really going toward doing anything. It's just being burned so that the temperature can raise. And in fact, most of the energy just gets wasted. It doesn't actually go toward anything. So that's IL-1. IL-1, and in fact, a lot of these cytokines, they don't only act on other cells, they act on the cell that made it. So IL-1, when it's secreted by the macrophage, it doesn't only act on cells that are around the macrophage or even distant from the macrophage. It also acts back on that same macrophage that just secreted it. And when that happens with IL-1 and with tumor necrosis factor, that causes the production of the cytokine IL-6. And IL-6 has important functions that I'll talk about in a moment. And then we have tumor necrosis factor alpha. These two cytokines have almost the same activity. They do almost the same thing. They're very slightly different, but they do almost the same thing. Okay, so tumor necrosis factor alpha, this is an inflammatory cytokine. It activates the vascular endothelium, increases vascular permeability by causing endothelial cells to contract, and therefore the gaps between the endothelial cells open up, and that allows blood fluid and blood proteins to leak into the tissues where they can have an effect on the infection. For example, you're leaking vitamin B12 binding protein into the tissue, where it's going to help tie up vitamin B12 so that the pathogen cannot get that growth factor. And you're also releasing other serum proteins into the tissue when you increase your vascular permeability. So you have an increased entry of IgG antibodies, complement proteins from the serum, and cells from the blood so that they can easily get into the tissues. And when you start draining all those fluids from the blood into the tissues, that fluid has to go somewhere. And what it's going to do, all that fluid is going to flow through the lymphatics, and it's going to drain to lymph nodes. And those lymph nodes contain T cells and B cells that will survey the lymph for antigens. And if there's foreign antigens present, then you'll get an adaptive immune response to them. And eventually, all of that lymph is going to flow way back into the bloodstream, and we'll talk more about that later. But eventually the lymph does make its way back to the bloodstream, but it has to pass through lymph nodes first. It has to pass through a lymph node first, at least one lymph node. Systemically, tumor necrosis factor is fever-inducing, so it acts on the hypothalamus, causes prostaglandin E2 secretion, which increases temperature set point of the body. Mobilization of metabolites, meaning it generally promotes the production of other inflammatory mediators. And actually what this really refers to is the fact that you're mobilizing fats and you're mobilizing carbohydrates and proteins. You're burning all those things so that the temperature set point of the body can increase. Okay? And tumor necrosis factor is also a strong promoter of shock. And in addition, tumor necrosis factors, let me compare and contrast these two for a moment. Most of their activities are identical. The only difference is interleukin-1 is better at getting you fever. Tumor necrosis factor is better at causing inflammation, and it can also cause coagulation. So it can cause clotting in the local proximity. And clotting helps to prevent the spread of the pathogen through the bloodstream, essentially. Okay, and tumor necrosis factor is more potently inflammatory than IL-1, but IL-1 is a stronger fever inducer. And then we have IL-6, interleukin-6. IL-6 is not really directly inflammatory. It doesn't really act on endothelial cells. It doesn't really act on smooth muscle cells. It acts on other cells. So it acts on lymphocytes. It can act on naive T cells, and it can cause a Th17 differentiation. And it acts on B cells, and it helps activate B cells so that you get increased antibody production. This is important because a lot of those plasma cells that you a lot of those plasma cells that you produce during the immune response, they actually traffic to the inflamed tissue where they secrete their antibody. B cells may traffic wherever they want to, but a lot of them go to the inflamed tissue to secrete their antibodies once they've encountered antigen. And so the IL-6 can even make them better at secreting antibodies. IL-6 is not only important for its local effects on these two things, but also its systemic effects. It's a strong inducer of fever. And you could say that IL-6 is probably one of the more important long-range cytokines because it has an extremely long half-life. We're talking an hour or two. So it stays in the blood for a really long time before it's degraded. These other ones, they only have a few minutes for a half-life, so they don't have as strong of a systemic effect. IL-6, though, very strong systemic effect, can easily act on distant tissues. And it can induce fever. It can induce acute phase protein production by the liver. So basically what happens is the IL-6 is secreted by activated macrophages in the tissue. It makes its way to the blood, and it circulates through the blood until it reaches the liver. And it binds to IL-6 receptors expressed on hepatocytes in the liver, which are just the liver cells, basically. And those hepatocytes begin making proteins that are upregulated during acute inflammation. So they begin making proteins like C-reactive protein, vitamin B12 binding protein, transferrin, lactoferrin, etc. All of those proteins help in the immune response. All of those acute phase proteins, they're going to help mark pathogens for destruction, and they will sequester vitamins and minerals and such from pathogens so that they have a harder time replicating in our body. Okay, so that's IL-6. We'll come back to like all of this at least once, if not more than once. And then we have IL-8, also called CXCL8, because it is a chemokine. And this is a typical chemokine nomenclature. The reason it's called CXCL8 is because there is a motif that is repeated in the protein's structure that is conserved across several different chemokines of this class. And it contains a cysteine followed by some kind of amino acid, followed by another cysteine by a leucine. And that motif is present in this protein, and it's also present in a couple of related chemokines. And the job of IL-8 or CXCL8 is to recruit neutrophils, basophils, and T cells from the blood to the site of infection. So it is a chemotactic factor. What that means is it directs the movement of those cells. Okay, they're going to move toward the IL-8. They're going to move toward a higher concentration of IL-8, and they can tell the gradient and where it is. They're pretty good at that. Lastly, macrophages make interleukin-12. This is a cytokine with two activities that you should care about. First of all, it is a strongly activating natural killer cell cytokine. So it binds to receptors on natural killer cells, and it causes them to become activated. They get all angry. They become really good at killing, and they begin making more cytokines. They make interferon gamma, which in turn can act on macrophages and make them angry. And that serves as a feedback loop between macrophages and natural killer cells. Macrophage makes IL-12, which activates the natural killer cell. The activated natural killer cell makes interferon gamma, which activates the macrophage, and it goes like clockwork in a positive feedback loop between these two cells. Secondly, IL-12 induces the differentiation of naive T cells, CD4 positive T cells, into Th1 cells, which helps promote the antiviral immune response. Okay, Th1 cells, they're good against viruses, and they're good against intracellular bacteria and other intracellular pathogens. You can't kill it from outside the cell because the pathogen isn't outside the cell. So you have to destroy the reservoirs of the pathogen where the pathogen is replicating, i.e., you're probably going to need to kill a bunch of infected cells. Th1 cells help promote that response. So, a little bit more elaboration on tumor necrosis factor alpha. Macrophages are not the only cells that make TNF. Monocytes and dendritic cells also make it, and that shouldn't surprise you because those are the cells that are most closely related to macrophages. And they make it under the same context. They've encountered a pathogen or they've been stimulated with some cytokine, and that causes the production of TNF. Th1 cells also produce tumor necrosis factor alpha, and they make it in response to T cell receptor stimulation. And natural killer cells make tumor necrosis factor. It's pro-inflammatory, which I've already explained. It promotes macrophage cytokine secretion, so it acts autocrine on the same macrophage that just made it, or it can be produced by any of these other cells to act on the macrophage. And in response to being stimulated with tumor necrosis factor, macrophages make more cytokines, and they become better killers. And it will also induce IL-6 production by macrophages, and IL-6 is a cytokine that comes a little bit later into the immune response. It's not made at first. You need some of these earlier cytokines to be made first, and they will act on the macrophage that's making them, and then you finally get the IL-6 probably a day or two into the infection. TNF activates natural killer cells, which I kind of explained. It leads to fever. It promotes blood coagulation or clotting by increasing the procoagulatory protein synthesis by endothelial cells and inhibitoring the synthesis of anticoagulatory proteins by the endothelial cells. And this implicates tumor necrosis factor in the pathogenesis of disseminated intravascular coagulation, or DIC. That is a topic that we'll talk about a little bit later on. Just know that it involves clotting, and a whole lot of clotting, and tumor necrosis factor can lead to it. In chronic contexts, tumor necrosis factor helps to promote the formation of an immune structure called a granuloma. More on that later in this slideshow. And it can promote angiogenesis. It can promote the what do I want to say? The growth of new blood vessels. Okay, that is what angiogenesis is, the growth of new blood vessels. So that's sort of an interesting, unexpected activity that it can have. Next, we have interleukin-1. The major form of interleukin-1, there's a couple of different forms. The one that we care about is interleukin-1 beta, which is like an isoform of it. And interleukin-1 begins as a product that has to be cleaved in order to become biologically active. The synthesis of interleukin-1 is upregulated by pattern recognition receptors, can be upregulated by cytokines and other inflammatory stimuli. It is pro-inflammatory, just like tumor necrosis factor, and it activates macrophages in a similar fashion to tumor necrosis factor. So it can act autocrine on the same macrophage that made it. It can promote CD4 positive T helper cell co-stimulation during antigen presentation, which means that it is what we would call a danger signal. Danger signal is just another word for co-stimulatory signal. Okay. At very high levels, IL-1 will act on cell hypothalamus and cause fever. IL-1, it turns out, is really important for fever. It's even stronger at producing fever than tumor necrosis factor, and it may be the body's most important inducer of fever. The main sources of interleukin-1 are macrophages, monocytes, and dendritic cells, but it can be made by some other cells, and that is important. Fibroblasts and epithelial cells can both make it, and these serve as some of the earliest sources of interleukin-1. So they're going to realize that there is an invader, and they're going to become activated and release interleukin-1, which signals to nearby macrophages that there is a problem, and that is a very early signal that there is a problem. For example, if a pathogen is infecting the intestine and begins damaging the intestinal cells, those intestinal cells will make interleukin-1, which helps to activate macrophages. And that gets the whole process going. Then we have IL-6. Interleukin-6 has a long half-life relative to IL-1 and TNF, and therefore it stays in the blood for a lot longer. We're talking multiple hours. IL-6 is secreted by monocytic cells in response to PAMPs being activated, or PAMPs activating PRRs. So basically in response to a pathogen being detected. And IL-6 is also made by Th2 cells. The long half-life means that IL-6 can circulate all the way to the liver and act on the hepatocytes to cause them to produce acute phase proteins like C-reactive protein, transferrin, etc. And here, in this table right here, we have a list of some of the acute phase proteins. Anything that's increased is going to be an acute phase protein. So proteinase inhibitors, coagulation proteins, complement proteins, transport proteins for certain nutrients, and miscellaneous proteins, which includes some of the C-reactive proteins, serum amyloid A protein. Most of these are proteins that are going to bind to pathogens and mark them for phagocytosis. And C-reactive protein can also activate complement. We'll talk more about complement later. IL-6 can promote B-cell activation, differentiation, and antibody secretion. IL-6 promotes development of Th17 cells from naive T helper cells, and it acts on the hypothalamus, causing fever. Okay, so fever is a common thing that all of these three cytokines do, but you can see that they're a little bit distinct from each other. They have preferred things that they're good at, right? Even though they have overlapping functions, they have things that they're good at and they're better at those things than other cytokines. And then we have IL-8, also called CXCL8, which is a chemokine or chemotactic factor. It guides neutrophils, T cells, and basophils from the blood to the infected tissue. IL-8 also helps strengthen adhesion proteins on leukocytes. It links integrins to the actin cytoskeleton, which helps promote tight binding. Tight binding is a required step for any white blood cell to leave the blood to enter the tissue. IL-8 is made by macrophages in response to pathogens, but it's also made by monocytes, mast cells, endothelial cells, and fibroblasts. And lastly, IL-8 can also help promote angiogenesis, or the growth of blood vessels. And then we have IL-12. It's produced by macrophages and dendritic cells. It promotes Th1 differentiation from naive CD4 positive T cells, and it activates natural killer cells. Okay, and this figure shows you a couple of the things that the macrophage cytokines are involved in promoting. Notice that they do have pathological effects. They don't only have protective effects, like the recruitment of white blood cells to help fight the infection, the production of more leukocytes from the bone marrow, the production of acute phase proteins, promotion of fever, sleep, and anorexia. All of these things are thought to be protective effects, right? But pathological effects also result from the production of pro-inflammatory cytokines. For example, tumor necrosis factor can reduce the output of the heart. It can make the heart less contractile, and it can help promote shock through that regard. It can act on the blood. Well, specifically, it can act on endothelial cells and cause them to promote coagulation. And coagulation can result in the formation, or does result in the formation of blood clots. That can be good because it can help prevent bacteria from getting into the blood from the tissue, but it is also potentially bad because if you have a really big blood clot in your blood vessel, that clot can detach and circulate to wherever it wants to go. It may end up in your lungs, it may end up in your brain, and it may end up in your heart. It can cause a heart attack, a stroke, or pulmonary embolism, or pulmonary thrombus. All of those things can cause you to die. So that can be a rather negative side effect of tumor necrosis factor, especially at high levels. And in addition, tumor necrosis factor has been shown to induce insulin resistance in skeletal muscle. This can help promote type 2 diabetes. And it also causes cachexia, or the wasting away of stored nutrients. So you begin losing all the fat stores that you have, you begin losing all of the protein stores that you have, and the lipid store, or excuse me, the carbohydrate stores that you have, and you begin to look rail thin. Tumor necrosis factor can promote that, especially over the long run. If you have a chronic inflammatory issue, this can happen to you. Okay, these things can happen. Usually the longer it happens for, the more tumor necrosis factor you have, and the longer you have it being made for, the worse off you're going to be. Okay. And then this slide you'll see again, but it shows you that IL-6 helps to promote a bunch of different proteins being made by the liver, including an important protein called C-reactive protein, which binds to bacteria and marks them for phagocytosis. Okay. And another figure showing you a couple of the things that these cytokines can do. I don't think I have anything additional to say here. This slide shows you how pain can be promoted by inflammatory mediators. Notice interleukin-5, histamine, tumor necrosis factor, IL-1, IL-6, and IL-17. All of these things can actually act on neurons that sense pain in the skin and in tissues, and cause them to be more sensitive to pain signals. And so this helps explain why it hurts when you have an infection, why inflammation is painful, generally speaking. It doesn't explain the full story, but this can help us understand why that is. Okay, you can see that it is a little bit complex, but this is just an overview. You're obviously not expected to know all the different receptors for these or anything. Then we have these really important cytokines called the type 1 interferons. So this is, separate this from macrophages in your brain because these aren't necessarily related to macrophages. Type 1 interferons are made by a lot of different cells during viral infection. Type 1 interferons include 7 proteins. All of them start with interferon. Interferon alpha and interferon beta are the two most important of the type 1 interferons, but we also have 5 others. Now, interferon gamma, you've heard me talk about a lot. It is not one of these. It is not a type 1 interferon. And in fact, it is what we call a type 2 interferon. And that is an important distinction for you to keep straight because it is completely different. So type 1 interferons, they are made by practically any cell in the body, any kind of tissue cell, downstream of signaling through toll-like receptor 3, 7, or 9, or intracellular pattern recognition receptors. So we have all of these different receptors that recognize viruses. All of these receptors are recognizing viruses. And all of them turn on interferon regulating factors. Okay? A virus component, such as double-stranded RNA or DNA that's in the cytoplasm where it shouldn't be, that's an indicator of viral infection, and that can act on these toll-like receptors and cause the turning on of interferon regulating factors, or IRFs. IRFs are transcription factors. Their job is to cause the transcription of type 1 interferon cytokine genes. Okay, so what happens is a cell recognizes a virus through these different receptors, right? And then interferon regulating factors are turned on, and type 1 interferons are made by that infected cell that recognized that virus. We also have these really cool cells called plasmacytoid dendritic cells, or PDCs, that are highly specialized to secrete huge amounts of type 1 interferons during a viral infection. They are capable of secreting more than 1,000 times as much as any other cell of these type 1 interferons. They make literally tons of type 1 interferon during a viral infection. And interferon beta has an important source in fibroblasts. Fibroblasts are an important source of interferon beta. Okay. Interferon alpha is made by tissue cells, plasmacytoid dendritic cells, or immune cells. So type 1 interferons, once they're made by an infected cell, they bind to a receptor called IFNAR, or the interferon alpha and beta receptor, on neighboring cells. That stimulates the transcription of interferon-stimulated genes, or ISGs. For example, an enzyme called RNase L is going to be produced in the cell. RNase L, the gene for it, is going to be transcribed by transcription factors that are turned on through the signaling pathway of the interferon receptor. And we also have a protein called protein kinase R that is going to be produced from type 1 interferon stimulation. And the job of RNase L is to degrade mRNA, and specifically it looks for viral mRNA, and it can recognize certain structures that are unique to viruses. And protein kinase R is going to turn off translation by turning off the eukaryotic initiation factor 2 alpha, which is a component of the ribosome. So it turns off the ribosome so that the cell stops making proteins. Not good for a virus. The virus needs the cell to be making a lot of protein in order for more viruses to be made. So if the cell stops making protein, well, the virus is kind of screwed. It can't replicate anymore. In addition, interferons acting on a cell will cause that cell to upregulate the expression of MHC1 on all cell types, and it will downregulate global cellular protein production. In addition, for professional antigen-presenting cells, your monocytes, macrophages, B cells, and dendritic cells, Being stimulated with interferons, type 1 interferons, will cause the upregulation of MHC2 and B7. So it can also piss off the antigen-presenting cells. Not only the tissue cells begin making MHC1 to show that they don't have a virus, you also begin having increased antigen presentation to T helper cells. In addition, type 1 interferons will act on natural killer cells and activate them. That makes them many times better at killing targets. 100 to 1,000 times better at killing targets. And lastly, type 1 interferons can induce fever or pyrexia. Okay, so very important class of cytokines here. Not one that really fits into any of my previous slides, but I kind of talk about them on their own. So that is type 1 interferons. And here we have three figures that I wanted to show you. Interferons acting on their receptor will induce an antiviral state in a cell. And that happens through two major mechanisms. Protein called something synthetase will cause the production of a really weird RNA molecule that is recognized by RNase L to activate RNase L. And RNase L then goes crazy degrading RNA molecules in the cell, leading to decreased protein synthesis by that cell, and therefore inhibition of viral replication. In addition, interferons will cause the production of protein kinase R, and protein kinase R phosphorylates the eukaryotic initiation factor 2 alpha that is required for protein synthesis, leading to inhibition of ribose and therefore inhibition of protein synthesis. Again, viruses do not replicate as well. And this right here is a list of all the things that the type 1 interferons can do, most of which I've already explained. And then this here shows you some of the timeline of an antiviral immune response. So interferons are really important in the early control of the virus, and they're going to spike after only about 2 days. So they're important to make sure that there isn't a whole lot of virus made early in the infection. Then natural killer cells swing in. They're getting activated by those interferons, and that causes them to get all angry. They're recruited, and they start to kill virally infected cells. And finally, the T cell response, it lags behind all of the innate responses. It finally kicks in, and they finish the killing of the virus. Okay, so you notice that the virus only really hits 0 once you get the T cell response kick in. All right. And this here is sort of like a web of the effects of different interferons, how they're made, and the specific proteins that are activated in response to interferons. But really what you should focus on here is the outcomes. Interferons will result in decreased translation, the upregulation of MHC1, inhibition of a cell to be virally infected to begin with, and that can happen because these proteins called the interferon-induced transmembrane proteins get turned on in response to the type 1 interferons, and they can actually inhibit the fusion of viral membranes with cell membranes, interestingly enough, meaning neighboring cells that were stimulated with those interferons that were made by the virally infected cell, they are less likely to ever be infected with a virus to begin with. So that can actually save them from even being infected. It leads to the upregulation of MHC2 and B7 on antigen-presenting cells. Interferons also result in fever and increased natural killer cell killing. Okay, so this is just a way for me to show everything that happens. And finally, you get to see the revealed structure of some of the antigen presentation molecules that I've been talking about. So we're going to move into the topic of T helper cells now. T helper cells, they respond to MHC class II molecules. And MHC class II is a two-legged molecule. It has 2 protein chains. And then it also has this peptide binding cleft that can bind big, long peptides greater than 10 amino acids in length. And then we have C1, which is a one-legged molecule. It only has one chain, the alpha chain. It associates with a protein called beta-2 microglobulin, and that serves as sort of a crutch for it to fold properly and not fall over on the cell surface. It only has one leg. If it tried to stand up on its own, it would fall over, so it needs the crutch of beta-2 microglobulin. And MHC1 molecules, they have a preference to bind to smaller peptides, usually 8 to 10 amino acids, although I think we go with different numbers in this class, but anyway. Now we have CD1, which structurally resembles MHC1, but functionally more resembles MHC2. It has only one chain, and it associates with beta-2 microglobulin. And what it does is it doesn't bind to peptides, it binds to lipids, or glycolipids or lipopeptides. And there are a very large number of pathogens, especially bacterial pathogens, that need CD1 to be present in order for us to fight them off. Okay, so CD1 presents lipids, MHC presents peptides to T cells. Okay, and this is another figure showing you the MHC proteins. Now in humans, so MHC is the major histocompatibility complex, and that is true for both the gene and for the protein. We can call them MHC. But in humans, we get a little more specific. We call it the HLA, the human leukocyte antigen. That is the human variant of MHC. Okay, we'll get more nitpicky about that for the next exam, but this figure just kind of shows you more detail on the structure of MHC 1 and 2. And you see that both of them have this peptide binding cleft at their very distal tip, and that is where T cell receptors bind to. They bind to a portion of the MHC molecule, and they bind to the peptide that has been loaded into the MHC molecule. Okay. And here's the structure of the T cell receptor and the B cell receptor. The T cell receptor for alpha beta T cells has an alpha chain and a beta chain, and it's associated with a protein called CD3. CD3 has a couple of different forms that look interesting. So this is CD3 here, this is CD3 here, and this is CD3 down here. So CD3 has an epsilon chain, a delta chain, a gamma chain, and a zeta chain. And epsilon can bind to delta or gamma, whereas the zeta chains pair together in a homodimer. All of these contain activation motifs inside the intracellular domain, and they basically allow the T cell receptor to signal to cause activation of the T cell once the T cell has encountered antigen. All right, so without CD3, the T cell receptor would not be able to signal. It would not be able to actually transduce the fact that it has encountered antigen to the cell. And then we have the B cell receptor, which on naive B cells is always a molecule of IgM, membrane-bound IgM. And IgM, when it's membrane-bound as a B cell receptor, and in fact all B cell receptors, they are Y-shaped proteins that look like this. They have 4 polypeptides. They have a heavy chain, which is this big long chain, a light chain, and they have 2 of each of those. Okay, and the antigen binding regions are way out here. And the B cell receptor associates with Ig alpha and Ig beta, so it can transduce its signal to the cell. Obviously, one cell is not going to be expressing both of these. It's either going to be expressing a T cell receptor or a B cell receptor, depending on if it's a T cell or B cell. Okay. So the antigen binding sites, they are the very distal tips of the proteins. These are CD3 molecules. And these yellow boxes in here, which I told you are activation motifs, they have a more specific name. We call them the ITAMs, the intracellular, I actually forget the name. I think it's like something activation motifs. Oh well, I forget the name. But basically, they allow for the cell to actually be signaled once the receptor has bound to antigen. Okay, Oh, I remember. It's immunoreceptor tyrosine containing activation motifs. Okay, ITAMs. More on that on exam 2 with signaling. I'm just sort of trying to give you an overview of all this stuff right now. Okay, so now we're looking really closely at T cells. And this is what we call the immunological synapse right here. The immunological synapse is basically the view of two cells of the immune system that are interacting. Usually, we're talking about an antigen presenting cell and a T cell of some kind. In this case, we're looking at a T helper cell that is being activated and it is naive. All right. And it's being activated, of course, by a dendritic cell, the only cell that can activate a naive CD4 T cell or a naive T cell of any kind. So the dendritic cell is expressing class 2 MHC molecules that has been loaded with a peptide antigen. And the T cell has a T cell receptor that binds to the antigen peptide inside the MHC molecule, and it binds to the MHC2 molecule itself. And then you also have CD4 that stabilizes the connection between the TCR and MHC2, and it binds to the outside of MHC2, which allows it to stabilize that connection, and it kind of just holds it in place in a way. And that signal will be transduced through CD3, which will allow the T cell to become activated. You also have adhesion molecules that are going to be localized outside of the T cell receptor MHC2 molecules. So they're going to be more on the periphery of the cell. You have LFA1 binding to ICAM1. These are not important details. You have, of course, the B7 signal. B7 expressed on the dendritic cell binds to CD28 on the T cell. That causes co-stimulation, and that is a required signal for the activation of a naive T cell. And then you also have cytokines being made by the dendritic cell to help drive the differentiation of the naive CD4 positive T cell. Okay, so depending on what these cytokines are, they're going to tell the T cell what kind of T cell it needs to become. All right, and this is a little bit more of a groovy view of it, I guess, or more of a protein structure based view, but it's essentially showing you the same thing. Signal 1 is the T cell receptor binding to MHC2. Signal 2 is CD28 binding to B7. And B7, there's actually two forms of it. There's B7-1 and B7-2. We just care that you know B7 though. But B7-1 is also called CD80, and B7-2 is also called CD86. So if you ever see those referred to as CD80 or CD86, just know that it's B7. And then we have the T cell activation of a B cell or professional antigen presenting cell. So the T cell is now a T helper cell. It leaves the lymph node, it circulates through the body, it enters the inflamed tissue, and it begins looking for MHC2 molecules that contain that specific peptide that it is specific for. All right, and when the T cell receptor binds to that antigen, it causes the T cell to respond with a heck load of cytokines. So the T cell is going to be making just a lot of cytokines. Those cytokines are going to act on the antigen presenting cell, for example, macrophages or B cells or monocytes, and it's going to activate and provide T cell help to that antigen presenting cell. All right, so it's slightly different. In the first case, the T cell is not getting cytokines to the antigen presenting cell. In the second case, it's providing a lot of cytokines to the antigen presenting cell, because the goal here is to activate the cell that has taken up antigen and is presenting that antigen. Okay. And then we have cytotoxic T cells recognizing a target cell. Effector CTLs recognizing a target cell. Again, you have a T cell receptor binding to an antigen, but in this case, the antigen is a peptide that's been loaded into MHC1. Remember, CD8 positive T cells respond to MHC1. And in this case, you have CD8 being expressed on the surface of the T cell, and that binds to the outside of MHC1 to stabilize the connection between the TCR and MHC1. It kind of just holds it in place and allows the activation to happen. And then, of course, you also have the adhesion molecules happening at the same time. Now notice that there is no B7 signal here. There is no B7. That is because effector cytotoxic T cells do not need B7 in order to kill target cells. We wouldn't want them to require B7 because they already received B7 way back in the lymph node from the dendritic cell. And chances are virally infected cells are not going to be expressing B7. So that would be kind of pointless for us to require cytotoxic T cells to, require B7 in order to kill the target. And when this T cell recognizes its antigen, because this is an effector cytotoxic T cell, it's going to kill the virally infected cell right here. Okay, so just another view of that. And this is a slide you've kind of seen before already. It shows you that naive CD4 positive T cells are activated by dendritic cells through the MHC2 and co-stimulatory signal. They turn into effector CD4 positive T helper cells, memory CD4 positive T cells, and follicular helper T cells that provide B cell help. And then this figure it's just showing you some of the co-stimulatory signals. The one that you really want to care about is B7. And B7 binds to CD28 on the T cell. B7 is expressed on the antigen presenting cell. CD28 is expressed on naive T cells. Okay, that is the major co-stimulatory signal involved. It turns out that it can be more complicated than that. That is the view that we want you to really have. But T cells can recognize antigen without B7. And so long as the dendritic cell has a molecule called CD40 on its surface, that is allowed. That is because when a T cell recognizes MHC2 loaded with its specific peptide, it begins to express CD40 ligand. CD40 ligand binds to CD40. That causes the dendritic cell to begin making B7, which causes the co-stimulation of the T cell. So it can be a little bit more complicated. Sometimes dendritic cells are not adequately expressing B7, but there are ways to circumvent it. Okay, so the CD40 signal, it's mostly thought to just enhance the T cell proliferation response. Okay, it allows better co-stimulation, basically. The difference is, when you knock out CD40, you can still get an immune response. You can still get a T cell response. If you knock out B7 from being able to be expressed, you cannot get an immune response. Okay, so if you have a mouse that either does not have a gene for CD28 or does not have a gene for B7, you literally cannot get an immune response to anything. An adaptive immune response, I should say. So that is a much more important signal than CD40. And so it's the one that we really want you to know. And this slide is actually talking about some of the co-stimulatory signals that are involved in activating T cells. I told you about the CD40 one. I told you about the CD28 one. Specifically, CD28 is a co-stimulatory signal, but it allows for the production of a cytokine called IL-2, which is a growth factor for the T cell. It acts autocrine on the newly activated T cell to cause it to proliferate and make 1,000 copies of itself. And IL-2 binds to a receptor called CD25. That is the high affinity IL-2 receptor. Okay. This is the reason that you really need this signal is because of these two things. It allows you to express the IL-2 receptor and IL-2. Therefore, you can get a lot of T cell proliferation in response to it. There's another protein called ICOS, and it has a ligand called ICOS-L or ICOS ligand. Honestly, no one cares if you know that one, so we can skip it. And then CTLA4 is one that we may talk about later, but I'm not going to talk about it right now. For now, if you honestly just know the B7 binding to CD28 thing, you're golden. And we'll specify what you need to know in the future. This figure is sort of interesting. It shows the protein expression in T cells that were just activated by antigen. So when a T cell is activated, it rapidly produces a transcription factor called FOS, and then it begins making IL-2, which peaks after about 12 hours, makes the IL-2 receptor at about 1 day, and it makes CD40 ligand at about a day and a half, and then you finally get cell division around day 2 to 3. Okay? So I just think this figure is interesting. There's honestly nothing really important to take away from it in addition. So a little bit more before we... Oops, it's already 7. Okay. Sorry, I guess I'll have to save this for next